species is outcompeting other species for resources, and so the one that's currently growing right now is winning.
Yeah, so that's something that could have happened is you've got one species that just happened to get a toehold and took over, and it started reproducing asexually as fast as possible, keeping everything else out. If it's an ascomycete, which is possible, then you're never going to get those black dots. If it is a zygomycete, how come you don't have black dots yet?
Because it's not stressed yet.
Yeah, it's just it hasn't had a need to sporulate.
So don't you want to put it in like a fridge for a little bit, stress it out, and then
try that out? See if you can stress it out. Toss it in the fridge just for a day or two. Not where it's going to freeze accidentally, but just pop it in there and take it back out and see what happens for a day or two. If you still aren't getting black dots at that point and it's still like totally covered with the white fuzz, take a bit of that fuzz, stick it on a microscope slide in a tiny bit of water, cover slip, look at it. You're looking for septa in the hypha. If you do see septa, what do you have?
Is it zygomycete?
Not a zygomycete. Remember, they're cenocytic. They don't have septa in their hyphae. So if you do see septa, then you're going to need to start over with a new slice of bread, different source of dirt. If you don't see septa, then you've got zygomycetes that just aren't sporulating, or chances are slimmer, but you could have a zygomycete that doesn't have melanized spores and you're just kind of missing it. It looks like white fuzz, but the sporangia are on there. So maybe take your scope and zoom all the way out to the 100x so you get a bigger picture, and look at the surface of your bread and look for white or clear sporangia. So little blob on top of a stalk sticking up. That'll be a
So it's just the
zygomycete
same
also.
thing, just minus the melanized color.
Exactly.
Okay.
The most common stuff in soil around here is going to be melanized, but there's a chance that you get a zygomycete that's not melanized.
Okay, thanks.
Mm-hmm.
Hey dear. Just wanted to let you know I got your message. Yeah, Omar's just downstairs hanging out with Panda, which is kind of nice. At least
I think you've got your mic on.
Oh, I do.
Anyone else having trouble with any of the labs? I know this can be tricky, and there's so many like variables that you know to look for and think about once you've learned about mycology, but we're still so new at this that you see the white fuzz and you're going, what's going on? But it could be these 4 different things happening, or maybe we need to stress it out a bit. That just kind of comes with experience and learning the life cycles of these groups. Try to put the life cycle into context of what you're seeing on your bread, and it starts making a bit more sense.
You want us to keep that bread mold on the auger until the end of the semester, right?
Yeah.
Okay.
You don't have to keep the bread around as soon as you
I
get.
threw that away. My wife was getting a little sick of it.
Yeah, understandable.
All right.
What about the number of ascomycetes on Earth? Everyone finding sources for that?
I'm personally having a pretty hard time with that.
Okay.
I've only spent, granted, I've only spent probably 30 minutes, but I've had a hard time finding articles that directly address how many species there are. I don't know if anyone else is in the same boat.
I found some on the Google Scholar thing, but I had to look. Nothing would be obvious on the, by the title, but
Yeah.
usually by reading the abstract or reading the first little bit, you'll find something you can use.
Yeah, there is no paper that's the title is, this is the number of ascomycetes.
I figured that'd be the case. So did I don't know who was just talking. Was that Jeffrey?
Yeah.
Yes, that was me, and that was also me that was being embarrassing talking.
So did you just basically look for articles that were related to the topic of Ascomycetes, and then you just looked at the abstract to see if they addressed that topic?
Yeah, honestly, I did. I typed in, like how many Ascomycetes and whatever articles came up. I found something in almost every single one that I looked.
Yeah. Maybe
Okay.
a good search term might be Ascomycete diversity.
Okay, I'll try that.
Yeah.
Thank you.
So I got a question about that.
Okay.
So if we come across, let's say hypothetically we come across, let's say 20 articles, but they all give us different numbers. Do we want to take the average of all those numbers? Because I'm assuming that some of them are going to be mentioned in two or three more papers, while some may not be at all. You see what I'm saying? Should we just get an average, or do you want the highest number we can find?
Now, this is the point of the exercise because you are going to get different numbers from different papers. I want your critical thinking put to the test. I want you to make a judgment call based on what you're seeing in the literature. Okay, so you're practicing being a scientist right now, and let's say you've got a question, how many Ascomycetes are there? You're going to go out and do a literature search like this, and yeah, it's going to take more than half an hour. You're going to spend some time looking through these papers, and you're going to start just making a list like, okay, this paper says this number, here's some of their methods. This paper says this number, here's their methods. And eventually, you get 10 or 20 of these kind of down, you start seeing a bigger picture, okay? You can start putting it together, and I want you to think about, are they all measuring this the same way? How would I measure it? Which ones of these seem more likely or less likely? Everyone's going to give me a different answer for how many Ascomycetes there are. There's no right answer for this.
Right. Okay, I see what you're saying. I'll go with that.
Okay. If you want to take an average, if that makes sense to you, you're going to have to justify it. Okay.
Will do. Thanks.
Mm-hmm. I know it's a bit open-ended for an assignment. You might not be used to that, but that's what research actually is. There's no right answer at the end of the tunnel for any research project. We're grasping in the dark, looking for stuff. Yeah.
As far as the lab assignments go, I have one more question. Sorry.
Sure.
Is there
no problem.
anywhere on Canvas or on the website that just has like a description of each? Because sometimes I have a hard time like remembering, like keeping them straight, what we're supposed to be doing for each one type of thing.
If you click on the assignment in Canvas, it'll give you the instructions and what I'm looking for in your upload.
Okay, thank you.
Mm-hmm. I'll go ahead and share my screen.
I got another quick question.
Sure.
Are we going to have another, I guess, lab assignment coming up pretty quick?
Yep.
Okay. That's it.
This one's more fun if you like being outside and don't mind getting burned to death in wildfires or something. We are going to be collecting Basidiomycetes. So you're going to be wandering through the woods looking for mushrooms or wherever you can find them. And I want to go over that kind of right now. And so today our lesson is about Basidiomycetes, just kind of looking big picture, what they are and some of the major groups, how to recognize them. And then the assignment, where is it? But I've got it here in week 5. It's your fungal collections, but go ahead and get started on that. So let's talk about that. When I say macrofungi, I just mean not things like Sordaria that are hard to see. I'm looking for mushrooms, okay? So Chandler posted a picture here on Teams of some oyster mushrooms. So that'd be an excellent collection, okay? Just look for mushrooms, things grown out of the ground, out of dead logs, living tree dumps, whatever, okay? For that assignment, I want you to find 5 different looking big fungi, okay? And we're actually going to click here. It's in week 5. The mushroom characters cheat sheet, okay? That is this sheet right here. For each of your collections, I want you to make notes of all these characters. It's easier than it looks. There's the cap, here's stuff about the gills, here's the stem, whether or not it's got a veil, what that looks like. If you can find mycelium or rhizomorphs, describe those, and then where you found it. Right? Was it growing in big clumps, or was it by itself? Were they scattered? And where did you find it? What was the habitat like? What kind of trees were nearby? Was it growing on a root or in the soil? That kind of thing. This just is kind of a cheat sheet to give you all the ridiculous terminology. Okay, so cuspidate cap, that refers to this shape here. Is this making sense?
Is this like a classification chart that once you go through it, it tells you a species, or no?
No, we're not exactly doing that. So like a dichotomous key.
Yeah.
Yeah, so if you go to a dichotomous key, it's going to ask you like binary questions, okay? It's going to say the cap is convex or concave, right? And if it's convex, you go to page 22 and start there, okay? This is showing you what all those terms mean. So if your key says a papillate with nipple cap, well, you look in this cheat sheet here and you see, oh, that's this shape here, number 14. Okay. So yes, it does have that, or no, it doesn't. Okay. The problem with dichotomous keys is that you have to know all these terms in order to use one, right? What is a rugulose with fine wrinkles surface of a cap? Well, it's this. Okay. It's not going to give you pictures in a dichotomous key. I don't like using dichotomous keys for that reason. You've got to memorize like 400 different descriptive terms. Okay, the difference between floccose and canescent for furriness. Like, I'm not going to remember that, so I use a little cheat sheet like this. I'm not going to need you to do any keying out of your collections. I just want the description. Okay, so you're going to basically be making a Word document. This is all on Canvas for the Basidiomycete collection assignment. Make a Word document, throw in a photo, just cell phone snapshot of the mushroom where you found it, okay? And then maybe slice it in half, take a photo of that, and then take some of the spores or a piece of the gill, smash it on a slide, take a picture of that, okay? So a few photos and then description for size, shape, color, surface features, cap margin, right? The context, the odor and taste. Don't actually taste it. Just talk about the odor. Okay. Then describe the gills. This is where cutting the mushroom in half comes in handy. You can see how the gills are attached to the stem or not. Okay. So you slice it in half, take a look at the gills, then talk about the stem. Where's the location? Is it off center? Is it lateral? Is it central? Okay. So for each, I'm guessing like this, you'll be able to fit each organism that you find on a single Word document page. And so you'll be turning in a 5-page little document. 3 to 5 photos of the mushroom that you found, and then you can just number the descriptions. So you could do Roman numeral 1, A, B, C, D, E, F, and just give me the terms that describe it here. Okay. Should I do like a sample one for you to see this?
Yes, please.
Okay, I can make a sample.
Hey, Dr. Zong or
Yeah.
Chandler, do either of you have any recommendations like like can you point us in the direction of where we maybe should be looking for these?
Anywhere that's safe to go and not currently burning is a good bet. Up in the
Like,
canyons.
do we want to go next to a river or something? Or?
Yeah, you're looking for moisture. You can find these out in the desert too, but your best bet are just canyons around here. Aspen Grove always has great fungi. Provo Canyon, around Strawberry Reservoir, there's great fungi. Unfortunately, Utah is like one of the worst places in the world to find fungi, but they're still out there. It's just not like, if you went to Washington, you'd be tripping on mushrooms, but here you do have to look a bit closer. Look for rotten logs, moist, shady areas, near streams, underneath trees. Definitely not out in the open under sagebrush. You're going to find very, very few mushrooms under that.
Of course.
Are
Okay.
you gonna
thanks.
find
So-
different types of mushrooms at different elevations?
Yeah, you'll find different mushrooms at different elevations. And in particular, the plant species that are around, like the dominant tree cover, is going to have a big role in what kind of fungi you find. Because so many of these fungi, especially these like charismatic mushrooms, are associated ectomycorrhizally with different tree species. So aspens are going to give you different stuff than pine trees. Mhm.
Dr.
was
Zong,
gonna
could you elaborate on what makes Utah a bad place to find fungi? That sounds really interesting.
It's pretty dry here, and that's the main thing. And we have really long, cold winters in the alpine areas, and so that even after snow melt, we'll get like a little flush of mushrooms, but it lasts, 2 weeks in the spring. And then right about now, we start getting more mushrooms as the temperature starts dropping. But fall doesn't last very long in Utah, especially at high elevation where all the trees are. And so you've got like a 2 to 3 week window for the most part to find like these big, fancy, fleshy Basidiomycete mushrooms. It's not like fungi are rare here. It's just charismatic mushrooms tend to be less abundant than in areas where there's rainfall. Does that answer it for you?
Yeah, that's
Okay.
interesting. Thank you.
Yeah. I mean, there is the Utah Mycological Society is quote unquote amateur mycologists. A lot of them are excellent mycologists. They just don't do it professionally for a living. They go out usually once a month, except in the winter, on mushroom forays. I can link you into them. It's like $5 a year to join, and they'll take you out mushroom hunting. Pretty good group of people, usually like 10 to 15 people are showing up to these forays. You spend the day kind of walking around. Everyone brings back the stuff they find, and then they help identify it. And then if you found something edible, you can eat it. But having participated in these groups in other states where I've lived, the difference is quite stark in terms of how many mushrooms get found. Here it's some. In Oregon, it is like metric tons of mushrooms. And so it's just a matter of rainfall. Okay.
I had one quick question, wondering if anybody else had an issue with when I was trying to like study for week 4 stuff, the hyperlink was just taking me to the same place on a lot of the
Which
resources.
here?
Basically all that.
Yeah, I had the same problem.
Oh, crap. Okay, well, I will fix that. Oh. Sorry.
You're okay. I just wanted to make sure that wasn't on purpose. I didn't think it was. But.
Nope. It's always something. Making websites is hard. Okay. I will get hyperlinks fixed. You didn't miss anything super important by not being able to click on those because those are all to be found in the slides also. So as long as you got the slides to work, and I think that was working. Yeah. Okay. So let's chat about Basidiomycetes. All right. So Basidiomycetes are the famous fungi. This is what people think of when they think of mycology. Even though they don't think of all of these, almost all of the big fancy mushrooms are in Basidiomycetes. If people are looking for morel mushrooms, though, which actually can be found here after fires in the fall, so that might be worth your time. Those are actually Ascomycetes with apothecia. So other than them, for the most part, most of your charismatic mushrooms are going to be Basidiomycetes. So we're just going to check out kind of what makes them what they are and look at their life cycle, how it's similar and different from the Ascomycetes. Basidiomycetes evolved from Ascomycetes. Okay, so they're the most recent. They're kind of like the flowering plants of the fungus world. And they've really diversified quite a lot recently. Okay. So these are the other dikaryotes. Just like Ascomycetes are named after having an ascus and ascospores, Basidiomycetes have a basidium with this that makes basidiospores. Okay, basidium refers to a club, and so this kind of looks like a club, I guess. So that's where they get the name Basidiomycetes. Okay. Here, from A to B to C to D to E, we're looking at a basidium. Okay, this is just a developing basidium. Sometimes, like in your textbook, they may call that the probasidium. Okay. You can see it's got a couple nuclei in it, kind of barely you can see that. And then they fuse, so it goes from dikaryotic to diploid. Okay. Then the basidium starts growing and it starts producing these little spikes. And that's where the nuclei move after meiosis, is into those. This is your typical, like, prototypical, I guess, basidium, where it's just one cell here. It's got 4 spikes on it. The 4 products of meiosis, those haploid nuclei, each one goes into one of those. It lays down some cell walls, becomes a spore, and gets flung off. Okay. So in the life cycle, Basidiomycetes have an extended dikaryotic stage, just like Ascomycetes, but it's actually even longer in Basidiomycetes. Because remember, in Ascomycetes, they can spend a lot of their time as haploid. If you remember that, like, the haploid hypha will start growing, and it can actually start going through this asexual cycle, and it can spend the majority of its life cycle, or its lifetime, being asexual. That's not nearly as common in the Basidiomycetes. The haploid mycelium is pretty short-lived in Basidiomycetes. So that's a major difference between them and the Ascomycetes. We've got a basidiospore that germinates. It's haploid. It grows into this really small, short mycelium. If it doesn't find a compatible mate pretty soon, like on the order of days to a week, it dies. Okay, it had its shot. Okay, so Basidiomycetes really count on a couple spores landing next to each other that are compatible mating types and meeting up early on. Okay, what happens after they meet up? Plasmogamy, not karyogamy. That's what makes them dikaryotic. So here you can see, it's kind of, yeah, you can see it, depending on how zoomed in this is on your screen. We've got the different mating type nuclei shown as different colors, red and blue here. The haploid mycelium meet up, plasmogamy happens, And then we've got 2 nuclei per cell section in the hypha. Okay, 1 from each. Once this happens, once the basidiomycete goes dikaryotic, big changes happen. First off, it's not going to die right away, like if it stayed haploid. Okay, it just can't survive very long as haploid. But it's now also capable of making a mushroom. You cannot get a mushroom from haploid mycelium. Okay, as soon as it's got those 2 nuclei, a bunch of genetic changes happen. It starts turning on different genes, and we can have these complex kind of multicellular structures, these fruiting structures here. So it starts making a mushroom when conditions get bad and it's time for sex. Okay. Your typical basidiomycete that everyone thinks about, just the gilled mushroom like you buy at the grocery store, looks like this. Zoom in on the gills, and that's where the basidia are, sticking off of the gills. Okay. Inside the basidium, that is where karyogamy happens. Then and only then. So it's very brief. It's a probasidium. It's got 1 of each nuclei in it from the different mating types. And right before meiosis, they fuse together. So they meet up and instantly undergo meiosis. So that's your basidium forming there. And they join up karyogamy and instantly undergo meiosis. And those nuclei get pumped into basidiospores, and the cycle repeats. Notice that there's not a little side loop here for an asexual cycle. There are a few basidiomycetes that have been known to do asexual reproduction. It's not unheard of, but it's not common enough and not important enough to make it into the diagram of the life cycle here. Whereas ascomycetes, they would have a little offshoot. So, oh, over here we can spend a bunch of time asexual. Basidiomycetes don't really do that. Okay. So let's take a closer look at Basidia. Okay, singular. What's that?
I got a couple questions about the.
All right, I will back up.
Yes. Two things. So are the gills like condensed hyphae then?
Yes.
Okay, that's interesting. And then the other thing would be, you talked about they turn on a bunch of genes once they go dikaryotic, right?
Mm-hmm.
Is there any like actual benefit to having 2 nucleus? Are they having like one nucleus is pumping out certain protein or certain RNA and the other nucleus is pumping out? Are they like sharing the load, or?
It's more of a sharing the load kind of thing. So you're going from 1 nucleus pumping out a bunch of transcripts, right, to 2. And so it's a kind of like a critical threshold that gets reached.
Okay.
Yeah, and so they're making the same stuff, but once there's a critical threshold, it's maybe analogous to like quorum sensing in bacteria. Once you get a certain concentration of chemicals, it turns on other genes, turns some off. And so it's definitely a concentration gradient that's happening inside the cell. All right, any other questions? Moving a bit fast here. All right. Well, this top figure may kind of help illustrate the fact that a gill is just condensed hyphae, right? It's a mat of hyphae hanging out, okay? Everything in the mushroom, even the stalk, that's just hyphae also, okay? The mycelium itself is hyphae, the stalk of the mushroom, the cap, even the spores technically are just bits of hyphae that have been walled off with a single nucleus in them. Right? So hyphae make up everything you see in the mushroom. Okay, it's about 90% water, so if you dry it down, that's why they get really small and shriveled. This is kind of a cross-section of the gills here. The surface of the gill, what do we call that? It's got the same name in ascomycetes. The surface where the...
Lamellae or something?
Ooh, close. You're on the right track. Starts with an H. the sexually
Amenium.
hymenium, yeah, the sexually productive area where meiosis is going to happen. So that's our hymenium, is actually the surface of the gills. And so this is the surface of a gill, and you can see basidia just kind of sticking off of it. If you buy button mushrooms at Walmart or something, the gills are kind of white, but if you leave them in your fridge too long, they start turning black. Okay. That's just the spores developing. So you started with a young mushroom, the spores weren't ready. A lot of basidiospores are melanized, dark colors, and so you leave it in your fridge long enough, that's the basidia actually start forming. They're undergoing meiosis in your fridge and making spores. So that's why it turns like a powdery dark color, okay, along the hymenium. Okay. So this is showing this process down here with all the arrows pointing to it. What happens? All right, so let's start with our two. So we're dikaryotic. They come together. Meiosis happens immediately as soon as karyogamy. We got our 4 products of meiosis. Those move up, get pulled in with spindle fibers into the new spores that are formed, and then they're ready to go. Okay. There's a bunch of terminology here. We've got the basidium. That's where karyogamy happens and meiosis happens. The little antler shapes are called sterigma. Okay. And then the basidiospore is what forms on top of the sterigma. This is idealized, obviously. Over here on the left, the little video that's running is slowed down almost 1 million times. So it's really fast camera they use, and that's a basidiospore being launched off of a basidium. Okay. It is currently thought to be the highest acceleration of any object in biology, is a basidiospore being launched. They don't go very far. You can see it's not moving very far before it slows down, but it goes from 0 to like 30,000 Gs in less than a millisecond. Okay. Just gets launched way off there. All right. The final thing we haven't talked about pointing here is the clamp connection. You only see clamp connections in Basidiomycetes. You won't find them in Ascomycetes. You definitely won't find them in Zygomycetes because they don't even have septa between their cells. Clamp connection is how a lot of Basidiomycetes maintain dikaryotic, 2 different spores in each cell. So as it's growing, mitosis divides both of those nuclei, right? It needs to lay down a new cell and little divider. It divides both those nuclei. One of them gets put up into this little backwards facing piece of hypha. Okay. The other one just gets left behind. The cell wall gets laid down. Okay, that septum gets laid down. And then this little clamp connection comes back, merges with the previous cell, and the nuclei gets thrown in. All right. If you see clamp connections under a microscope, you know you're looking at a Basidiomycete. Not all Basidiomycetes have them, but only Basidiomycetes have them. Can you find the clamp connection in this micrograph?
Is it kind of at the top there? I don't really know how to describe it, but I kind of see it like in the middle of the screen, just a little bit. Yeah, right there.
That's it. Yep. So you can see the septum here. You can see it kind of comes off at a little angle like that. That's what we're looking at there, and there are 2 nuclei in that cell. Okay. So if you see those little bumps on your hyphae, then you can be guaranteed that you're looking at Basidiomycete. If you see that on your bread mold, for instance, you definitely don't have an Ascomycete or Zygomycete. You've got a Basidiomycete. Okay. In which case, leave that bread around and see what mushroom grows off of it, because it might be interesting. Okay. This is just the way that basidiomycetes maintain that dikaryotic state. Okay. Questions about that? Definitely something different from the ascomycetes that we've seen. All right. 3 main groups of basidiomycetes. The Puccinio-Mycotina, the Eustologino-Mycotina, and the Agaricomycotina. Agaricomycotina is what you're thinking of when you think of mushrooms. Okay. Eustologino-Mycotina are smuts. So like the junk that gets on corn and turns it into wheat lacoche for tacos in southern Mexico. These are plant pathogens, typically thought of as harmful for agriculture, unless you want to make smut tacos, which are actually quite delicious. And then the Puccinio-Mycotina, we've got some jellies in there, but largely these are the rust fungi. And these are very important plant pathogens. Okay. Agaricomycetes are the quintessential classical fungi. They got gills, they got a cap, stem, they make spores sticking off of basidia attached to their gills. Okay. But not all basidia are formed alike. We saw here an idealized version of a basidium. Okay, it's a single cell. It's got exactly 4 spores coming off of it because that's what meiosis makes. When we look at the actual morphology of fungi, we used to divide it up into homo and hetero basidiomycetes, whether or not it was a single cell or whether it was split up, fragmented. Okay, so turns out that's not a good group, or not a good character to use for grouping. Once again, we did not do a great job early on in mycology of picking characters morphologically. So these are kind of spread out all over the place, but we've got several different looking basidia. You can see Holobasidium, Heterobasidium, they're spread through all these groups here. Okay. Agarics, boletes, polypores, gastromycetes, they're some of the coolest looking ones, I think, and the russulas. Okay. I bet if you go out into the woods right now, you're going to find polypores hanging out on living or dead tree trunks. Okay, oops. I bet you're going to find under pine trees some boletes, maybe some agarics, and definitely some russulas. Because they're really closely associated. They're ectomycorrhizal, closely associated with pines. So let's take a look at the boletes. Instead of gills, these tend to have pores. Okay, so if you find a mushroom that has like a sponge surface underneath instead of gills, you're probably looking at a boletales. Okay. Of course, we've got stuff like G here. This is also, phylogenetically, it trees out with the boletales. Okay. Chorogonthus here, but it actually has gills. Okay. What would you think is the basal condition? The quote-unquote primitive state, gills or pores? Let me bring up the phylogeny.
Oh, I mean, just off of those pictures, it looks like most of them have gills, so gills, but...
Well, in these photos...
Well, actually, yeah.
The agarics and the russulas are the only ones that have true gills.
Flip that around then.
Yeah. Okay. Highlighted in blue here are the Homo basidiomycetes. So you can see that they're kind of like scattered all over the place. These are the boletes. See if we can find them on here. Where's the Bolatales? It's down here in Agaricomycete. There's the Russelales, those have gills. Polypores don't have gills. Dacromycetes don't have gills. Tremella doesn't have gills. We're thinking you can go maybe either way, but it looks to me, at least, and some other papers, that not having gills was kind of the first development here when Basidiomycetes split off from Ascomycetes. Okay, gills seem to be a rather newer development. Okay. All you need is a hymenium, a place for your Basidia to grow. Gills are one way of doing that, and they seem to be more recently evolved. Okay. But it also seems like it's been independently evolved, because here they are in the Bolatales. All right, so keep an eye out for these when you're out collecting mushrooms. There's a pretty decent chance that you'll find these. Some of them are highly prized edibles. So the porcini mushroom is a bolete. If they've got red gills, they're definitely poisonous. And some of them will make you sick anyway, whether or not they have red gills, but probably not kill you.
So are any mushrooms poisonous to the touch, or
No.
are they all ingested?
Yeah, just don't eat anything.
Okay.
Yeah. There are only a few types of mushrooms that I can recommend eating because they don't have any poisonous lookalikes. And one of those is like the oyster mushroom. Once you see an oyster mushroom, you know what they look like. Some people may have allergies, so if you want to eat oyster mushrooms, start with a small amount. But if you're fine going to the Asian market and buying some oyster mushrooms and eating those, then you're probably fine picking oyster mushrooms out in the woods, unless they're on a poisonous tree. Like some of them will grow on hemlock, and that can make you kind of sick.
What about around here? Like, what's good to find in Utah?
Oyster mushrooms, actually. They can be found on dead logs in moist areas, and they can actually overwinter. You can freeze them, and they'll kind of spring back up after it thaws. And so they tend to do pretty well in places with hard winters. Oyster mushrooms, I found them here. They're pretty easy to identify. Nothing else looks like them. They smell pretty unique. Once you've smelled them, it's kind of like a sweet, earthy smell. And they're relatively safe to go eating. I can't, in good conscience, say definitely eat any oyster mushroom you find. But if you've got a good ID book and you feel pretty confident, that's like your safest starting point for collecting edible mushrooms is the oyster. And we'll see them here in a second. Then we've got the gastromycetes. These are some of my favorite just because they're so weird. Okay. Gastro refers to like a digestive system, right? Kind of like a stomach. So these have spores that aren't forcibly discharged, and in a lot of cases, they're actually surrounded by mycelium. Okay, so your puffball. This is a puffball here in the lower right corner. You cut it in half. All the spores are on the inside. Okay. If the spores are purple, it's poisonous. Don't eat it. Okay. We've got the bird's nest fungi. Okay. We've got these crazy things, the stinkhorns. A lot of them put out these crazy nets. The spores are born in sticky masses on the top. Okay. We got earth stars, all these weird mushrooms where the basidiospores mature inside the basidiocarp, and they're not forcibly discharged. So if they're not discharged and they're stored inside, how do they disperse?
Something eating them?
Okay, so something could eat them.
Would it be rainfall?
Rainfall. When you were a kid, did you ever find a puffball and stomp on it? Is that just
Yes.
me? Yeah, I think that puffballs like co-evolved with human children because they're just so fun. You smash them and spores go everywhere. And so they actually evolved with rainfall. So here you can see a little hole gets formed right at the tip. It's almost like a little parathecium, okay? And when it's disturbed, the spores in massive numbers just get ejected, okay? The stinkhorn things, like these stinky squid mushrooms here, which are beautiful, crazy alien looking, but man, they are really stinky. You can smell these long before you see them. They're dispersed by like carrion beetles and flies and things because they smell like rotting meat. Okay.
Sorry, Dr. John.
Yeah.
Is this still under the subphylum of Agaricomycetina?
I'm sorry to report that these are paraphyletic. They're all over the place. But the ones we're looking at now are in Agaricomycetina.
Okay.
Yeah. We find these types of fungi, the gastromycetes, kind of all over the place. So here they are in the russeloids. Here they are in the agaric, like euagarics. They're in the boletes. They're in the gonthus clade. So they are spread out all over the place. Like your truffle, you can see here, number 7. Truffle, hypogeous, meaning it matures underground. It's found in the boletes, the gonthoids, the russeloids, and the euagarics. Okay. The ultra fancy, like black truffle, you know, you go to Smith's and buy some black truffle oil. Okay. That is here in the boletes. All right. Now we've got the agaricales. All right. These are traditionally the guild fungi, and that's how we used to classify. All guild fungi were called agarics. And so you'll still see that term. Agaric is kind of the slang term for this euagarics, the monophyletic grouping. Oops, found right here, the euagarics. Okay. Here we're seeing typical euagarics from your textbook. They've got gills here pictured in letter N. So this is a cross section of gills. The basidia are sticking off the side. We're used to these. These are like our standard mushrooms. Another thing you'll find out in the woods on dead tree limbs, rotten logs, are these heterobasidiomycetes, these fragmented basidiomycetes, often just called jellies. Okay. The auricularia, so named because it looks like an ear. The common name is the tree ear. This is another one that's actually good to eat and there are no like poisonous jellies that I know of. Most probably wouldn't be very palatable, but the auricularia is common in Asian cooking, ingredient in soups. Quite delicious, actually. I feel comfortable saying that if you find a tree ear, nothing else is going to look like it, and you're good to go on taking it home and boiling it up in some soup. And you might be pleasantly surprised. It's kind of crunchy. Okay. We call these heterobasidiomycetes because their basidia are segmented. Okay. It's tough to see here in the auricularia, but here's the basidium, and the sterigmata, the little antlers that branch out that contain the basidiospores, are really long in here, and there's a little line kind of coming down here showing that it's segmented. So this basidium is actually 4 cells instead of 1. In the Tremolaceae, they've got the same sort of deal. It's easier to see the cell wall in this photo, I think, or especially in this one. It's the same sort of deal as the Auriculariaceae. And then finally, the Dacrymycetaceae, okay, Dacrymycetaceae being one of the orders there. These are named because they look like a tuning fork. They only have 2 sterigmata coming off of the basidium. All right, so how does that work, given what you know about meiosis?
They don't do meiosis, I assume, because if they're splitting up into 4 for meiosis,
It should be 4, right?
yeah.
So is it
Meiosis
mitosis?
gives you 4 haploid nuclei, but they've
Or are they
only
just
got.
getting rid of 2 of them and reabsorbing it?
Yep. So their nuclei do kind of what human nuclei do in a cell, right? So to make sperm or eggs, we wind up with 4 nuclei in there. And what happens? One nuclei chemically kills the other ones, essentially. So it's like a little battle for survival. Same sort of things happening here. 2 of the nuclei get to survive, 2 get murdered and reabsorbed, okay? The common button mushroom that you buy at the grocery store also does this, okay? The Latin name for it is Agaricus bisporus. All right, so it's an agaric, it's standard guild mushroom. It's actually in the type genus for Agaricales. And bisporus, it's only got 2 spores. So instead of 4 sterigma on there, you only see 2. Okay. But they're not long and crazy like the Dacrymycetaceae here. All right. If you find a little yellow jelly like this out in the woods, there's a good chance that you will. Go ahead, bring it in, describe it, and then just with like an X-Acto knife or razor blade or something, cut off a super thin slice, stick it on a slide, and you'll see this tuning fork basidium on there. Okay. It's actually pretty exciting if you're a fungus nerd. I get excited by it. I can't help but collect those. And then I always collect the tree ears because I like to eat them. Okay.
Question.
Yes.
So these jellies are very different from all the other stuff we've looked at so far in Basidiomycetes. Is there any explanation of, I guess, why they look like this? Why they're so different? Is there any advantage to that? Or just kind of that's how they are?
I don't know. They're essentially doing the same sorts of things, right? They've got a hymenium, they're making basidia. Their basidia are a bit different in that they have extracell divisions. But, you know, at a fundamental level, they're actually not that different. If comparing them to like a standard mushroom, yeah, they look a bit different. But what about our gastromycetes? These are also quite unusual compared to a mushroom, and they're still doing the same sort of thing. They've got a hymenium, it's just inside of their body, so to speak. They've got basidia. They are dikaryotic right up until meiosis. So there's just a lot of ways to look while still performing the basic tasks of a basidiomycete. And so fungi, basidiomycetes have found a lot of different ways that work for them. There's really no constraint. These are all still, you know, hyphal filamentous fungi growing with a mycelium. That's 95% of their life is just a mycelium. So the sexual structures are kind of free to do whatever works for them as long as they're getting the job done. That'd be my hunch on this. And these are also, keep in mind, none of these like groups, Heterobasidiomycetes, jellies, none of them are monophyletic. So if we look at our little tree here again, okay, we see that we can kind of look at the numbers here, where are gills found, where are pores. toothed instead of gilled. The club fungi, whether it's a crust or gastromycete, and there's this spread all over the place. And so these forms have evolved independently a lot of different times. Okay. Rusts and smuts. This is the last major group of basidiomycetes we'll cover. They're the most annoying for biology students because of the life cycle. It's really ridiculous. The rust fungi require 2 separate living plant hosts. Okay. This is counterintuitive at first. We're gonna have to walk through this life cycle a few times, I think, before it kind of sets in. Let's start with our basidiospores over here on the left, because we know where those come from. They come from a basidium. What's their ploidy level?
Haploid.
Haploid. Okay. So we've got haploid basidiospores. In this case, we've got rust fungi are often named for the 2 plant hosts, and so this one would be the larch willow rust. Okay, they're specialized to those 2 species and nothing else. It lands on larch leaves. Okay, if it survives and gets a toehold, it will start growing into the leaf. Its hyphae will start digesting the leaf, or it finds a little opening, it goes in and starts sucking out nutrients from the leaf. It's definitely a parasite. Okay. These are haploid though, and so being basidiomycetes, they need to hurry up and mate real soon, or else they're not going to survive. Okay. But they can't grow very far, so one spore lands here on this left part. Okay, and it creates this little pocket, and that's basically as far as it goes. There's some hyphae that will stick out from this into the leaf tissue, but not much. And first thing it does is set this up and start making gametes. Okay. Over here is another mating type doing the same thing, drawn really close just to make this easier to visualize. Those gametes get passed over just kind of like pollen on a plant. Okay, so you've got a spermatium making sperm, you've got a spermagonia here that's kind of making all these gametes, and you get more or less analogous to cross-pollination. Then what happens genetically? When basidiomycetes mate, we get plasmogamy, but not karyogamy. So we get plasmogamy, we've got mating from a single cell into the larger one, and that starts triggering the dikaryotic stuff, right? So it's got it's crushed its threshold of concentration for certain chemicals and transcripts, and so it can start kind of really growing. It's hard to see here, but it starts making hyphae down further into the leaf, and it grows through the leaf to the other side of the leaf. Okay. At this point, this is all dikaryotic, right? This was haploid. Now it's mated through this kind of pollination mechanism, and once that's happened, it's gone dikaryotic, it grows through the leaf to the other side and starts making dikaryotic spores. Okay. This is pretty unique to the rusts. Dikaryotic spores here, you can see they've got both types of nuclei, a hollow and a filled-in circle. We call this an ecium, and we call these esiospores because they come from an ecium. Okay. They are dikaryotic. They go out into the wind or get carried by an animal or whatever, and they have to land on a separate host. In this case, they need to find a willow tree. Okay. When they land on the willow tree, they can start growing. They grow their mycelium, they infect the willow, and start making uridinospores, which are produced by what? What would we call the structure that makes uridinospores?
A
What's our-
basidium?
Nope. That would be a basidiospore.
What would this be? Just like,
The uridinium. Yep. It's once you get used to this, like it starts making more sense. In mycology, there's a separate spore name for a separate type of spore producing structure. A uridinium produces uridiniospores. So this will actually grow through the leaf of the willow, making it pretty sick in the process. And when it's ready to reinfect or move to a new part of the tree or whatever, it kind of just puts these out. It's almost like an asexual process. It can reinfect this willow tree over and over. Okay, and that's why it's got this little like loop back to the willow. These uridiniospores can only infect willow trees. If they land somewhere else, they're not going to survive. It needs that chemical signal from the willow. It needs genetic compatibility with it. Okay, it can keep reinfecting it, making uridiniospores. Okay. When it's ready to reproduce sexually, it makes a telium, which of course produces teleospores. Okay. The teleospores are where karyogamy happens and then immediately meiosis. Okay. So a teleospore technically is a basidium. All right. So a teleospore is a basidium. It's just a basidium that gets thrown off of the fungus. So this is a bit weird too. In your mushroom, your basidium is hanging out there on the side of the gill, and it makes basidiospores, and it throws the basidiospores off. In the rust fungi, keep in mind these are like a multi-billion-dollar-year industry problem. Okay, most research into mycology is into rust fungi. This is by far where most of the research dollars go. Okay. They throw not the basidiospore off of the tree, but the actual basidium. It can overwinter, and so it's got this really thick cell wall pictured here. In fact, a lot of them have to overwinter before they can germinate. Spring comes around again, so these hang out in the soil or wherever, and spring hits, and it grows its little basidium shape here, and we get our 4 basidiospores, which go on to infect the large leaves. Okay, so that's the life cycle. It needs both of these plant hosts, and it gets these special names, the ecium and the uridinium. Okay, so a lot of times you'll see it maybe in your textbook called like the ecial host. That's just referring to the host that makes the ecium. Okay. So pictured here on the right is to show you some of the crazy stuff that actually happens with rust fungi, particularly here, okay, in the ecial host, because we have to get gametes from one spot of the plant to another. Even better, gametes from one plant to another one for better like outbreeding. So how do they do that? This almost looks to me like parts of a flower, right? We've got anthers as receptive hyphae. We've got essentially like pollen grains here. We call them spermatia in fungi. We got to get some of this quote-unquote pollen onto a receptive hypha in another part of the plant or across the field somewhere else.
Are they being spread by pollinators?
Yeah, it's insects. So they're working just like flowering plants, right? They're having insects do their sexual reproduction for them, okay? They're getting the gametes together. So it works just like a flower. The analogy is almost perfect. It's got all the same sorts of parts. Insects are actually doing the quote-unquote pollinating or the spermatiating. I don't know what you would call that. Okay, they're moving these gametes to the receptive hyphae on another part of the plant or across the field on another one that's been infected. Okay. Pictured here at the top, you are looking at a fungus. That is the infected version of this flower here, down at the bottom. So these rust fungi are actually mimicking flowers. They mimic colors of flowers. They can even they can like trick the plant into producing tons of, what's the word? Sugary substance, really brain farting here. Nectar, thank you. They can trick the plant into producing nectar where it wouldn't otherwise to attract insects to help carry out this fungal reproduction. So they're hijacking the plant, they're hijacking plant chemicals, and they're kind of controlling it and tricking insects into spreading the fungal disease instead of the plant's own pollen. Okay. So that's ridiculous. Okay. Some of these will actually even put out pheromones that attract bees. Okay. So the bees come thinking that they're having sex with another bee, but they're not. They're spreading spermatium from a fungus. Okay.
So is it safe to assume that these pollinating insects are the only reason that this whole process is even successful? Because it seems way too complex for it to do without them.
Totally. Yeah. And these evolved with flowering plants, which co-evolved with insects. Yeah. This process is very complex. It's ridiculously complicated life cycle. It took me forever to get the hang of it when I was first learning mycology. I found it pretty annoying. Mostly it's the terminology that was messing with me though, and I let that get in the way. So when you think about it, it's just one of the plants that is its host makes the gametes for sexual recombination, right? So in our previous stuff, way back, where are we? All the way back here. We've got haploid mycelia that are just meeting up in the soil or in the tree trunk where they're growing, right? They germinate, they grow a little bit of mycelium, and hopefully they find each other in the dark, and hopefully they're sexually compatible, and so they can go dikaryotic. Now, here in our rust fungi, they need to accomplish the same thing. They just do it in a slightly different way. They break off little bits of themselves. It looks like a spore, but it's just a piece of hypha. They break that off and count on an insect to move it to a sexually compatible, same species, spermagonium. Okay. So yeah, without insects, this wouldn't work.
So are those little bits of hyphae considered gametes?
Yeah.
Okay.
Just like it may be counterintuitive for you, but like your typical button mushroom, that basidiospore lands on some cow dung and starts growing into a tiny little hypha. That hypha is actually considered a gamete also. Okay, because it's what's like donating the nucleus to the other receptive hypha. So here we're seeing the same thing, except instead of two kind of multi-celled hypha coming together in the soil, we've got two stationary little products, and we're breaking off bits and hoping they make it over to the other one. So yeah, insects are key in this. These rust fungi have co-evolved with flowering plants for so long that they're stuck like with their plant now, right? And so this particular species of rust has to have larches, and it has to have willows, and it has to have insects do the recombination for it on the larch, and then it's got to have a willow tree nearby, or else it's not going to be able to live.
Okay, so given that, these are also problems for crops, right?
Massive. In fact, probably the most economically damaging crop pathogen out there are these Puccinia fungi. Okay, they are wheat rusts, and they damage wheat, oats, rye, all those cereal grains.
So
You can imagine what a problem that is. Yeah.
why don't we just get rid of one of their necessary plants, like get rid of all the willows in the area? Boom, done. They can't.
You know what you've done? You have just re-engineered the solution that we actually try to use. So let me show you the cedar apple rust here. Apples are an important crop in Utah. There are actually quite a few apple orchards here, so it's economically important for our state. Cedar apple rust, as you might expect, requires apple trees and cedar trees. Okay, so before we knew about this life cycle, I mean, you can imagine how hard this was to figure out. It took decades and decades. We knew that like our plants were getting sick. We knew it was a fungus, but we had no idea that there was like this second host, okay? Because it looked totally different. When it's on the willow, it looks like this, and it makes these spores with 2 nuclei in them. And then it took us forever to figure out that those were actually Basidia and that they were overwintering, okay? And then it looks very different here in the larch tree, and we had no reason to assume that the things making the willows sick were these same things that we found in the larch leaves. Same thing was happening with apples, okay? So our apple trees, they're getting sick. They've got these little spots. If you take a little cross section of them, you can see down here in the right-hand side, this is the apple. There's these fuzzy bits on the top of the leaf, and then these like kind of gaping wounds on the bottom, okay? And until we figured out the entire life cycle of these rust fungi, we were doing something a bit stupid, and that some farmers actually still do because they haven't heard about this yet, apparently, is making windbreaks for our apple orchards out of cedar trees. It makes sense. Cedars grow really fast. They're dense. They make perfect little windbreaks. You don't want wind blowing through your apple orchard because it blows off the apples right at harvest time. They land on the ground. You lose a bunch of money. So most apple orchards in the United States were actually surrounded by cedar trees because they were a perfect little wind block. You can imagine the problem. We were setting up conditions perfect for this cedar apple rust. And so now the easiest way to get rid of rust on your apple trees is to burn down all the cedar trees within a mile radius. Yep. All right. For the cedar apple rust, which one is the teleal host and which one is aecial?
Teleal will be top, and then AC will be the bottom.
So teleal up here?
Yes.
Why?
So maybe I'm getting my terms mixed up, but so teleo is the one where it is like a flower, right?
No.
Okay. Whichever one's supposed to be like
Let me show you this.
Okay.
The part that acts like a flower is the aecial host because it starts on one side of the leaf. That's kind of like the flower analog. And then once it gets pollinated, so to speak, it's able to grow through to the other side and make aecium.
So that part is the bottom picture, I would say, because that looks more like a flower than just the rust on the leaves.
Ah, okay. I see what you're saying. Looking at this is the life cycle of the cedar apple rust here, this drawing. We see right over here, you really have to blow this picture up to see it better, but you can do that on your own time. We've got like the flowering part right here, the receptive hypha, the spermagonium. Okay, it gets quote unquote pollinated and then starts growing through to the other side, and that can reinfect the apple. So the flowering part in cedar apple is actually on the apple tree. Now, I'm saying flowering. I hope I'm not confusing you. It's not actually a flower, right? It's the spermagonium and the aecium. On the underside of the leaves, you can see like the little pockmarks that have come through. That's releasing those esiospores that can reinfect the apple tree. So it can go through this little reinfection cycle. Okay. It can then land on the cedar tree, and it makes these big old globs. It hijacks the little cedar berries, those blue cones on the cedar tree. Okay. Juniper actually is the genus, but it hijacks those, sends in some hormones, makes them grow humongous, and then puts out what essentially is like a standalone mushroom gill out of nowhere. So these little orange gelatinous things sticking out, that's pictured here. Okay, it's almost like a little gill cross section, and then poking out of it are teleospores. Those break off, land in the soil, overwinter, and then produce the basidiospores after meiosis, reinfects the apples nearby. Okay. How confused on a scale of 1 to 10 are you about the rust life cycle?
Pretty confused, but
Okay.
Yeah,
Better than when we started.
true.
How
Better
can
than we started.
So when we were on the previous slide, I feel like it all made sense. And then we moved on to the next one. And for some reason, I just feel like it doesn't look in how I thought it would.
Okay, let's see here. So on the previous slide, our spermagonia are at the top there. If we were to take this picture and turn it 90 degrees to the left, there's our spermagonium going in the surface of the leaf, and then eventually after they're fertilized, moving down to the underside of the leaf. So that's the equivalent of these two pictures right here. Okay. So that's the spermagonium moving and making esia. Those can go in and reinfect the apple. So that's like this part here, reinfecting. It can spread to the willow, which in this case is a cedar. Okay. So the spores landing on the cedar tree are dikaryotic. Okay, so it's landing on our willow. And then it's hijacking part of the willow and making teleospores. In this case, it's hijacking part of the cedar and making teleospores. So it's the same life cycle. It's just, I get that it's harder to see. The text is way small.
So the cedar is the one that has the basidium that's releasing spores to the apple tree.
Yes.
Okay. They grow
So
through the leaves.
on this orange glob, if you were to go into one of these with a microscope and look at it, you would see these little fuzzy, stocky bits sticking out of it, kind of like little hairs. Those are all teleospores. So these teleospores get knocked off, land in the ground. Okay. Winter comes, winter goes, springtime hits, and right as the apples start getting their leaves, their leaf buds on them, these teleospores get activated in the soil. They stick out this little tube here, the basidium, throw their nuclei into basidiospores, and those get kicked up by the wind or carried by insects or birds onto the apple tree. They infect the apple tree. So telium gets knocked off of this nasty blob, lands in the soil. Springtime, those spores come up, land on the apple tree, and start reinfecting it. So they can take over an apple tree in a matter of 3 weeks. They can go from one little leaf spot to the entire tree real fast because they can just kind of keep reinfecting it. Okay.
Does it kill the apple tree all the way,
Yeah.
or?
Yeah, it will. It's very possible. It certainly leads to diminishing crop returns. If you see these spots on an apple tree, an orchard farmer is going to freak out if they see this, because it means big money loss. Okay. There are things you can do. You can spray like fungicides on there and hope that helps. Right after winter, it's a good time to spray your soil with fungicides if you're hoping to prevent this, because that'll stop those teleospores from coming up out of the soil. But the problem with rust is that if you don't have cedar trees on your property, you can't exactly spray the soil for teleospores and hope that that prevents your apples from being infected, right? Half a mile away, somebody could be growing a bunch of cedars for a wind block, and that's where the teleospores are coming from. So you're kind of in a bind here. You can't go over to your neighbor and start spraying like horrifying chemicals on their soil and expect them to be happy with it. They're going to call the cops on you. And so this is like a constant problem because they have these two hosts. Their life cycle is actually really resilient. It seems like it'd be fragile because they depend on these two hosts, but something bad can happen to the local population of apples and they're still going to be fine, right? They can overwinter, wait for things to pop back up. You can come and spray your apple trees and kill off all these Esia all over them and maybe still eke out half a crop. And then next spring, they're right back because there's cedar trees somewhere in the area. And so instead of being really fragile, this life cycle gives rust quite a nice advantage. They have two homes they can kind of hang out in. If things are bad, they can stay there for a while. So honestly, the best way of preventing your apple trees from being destroyed by this rust is to get rid of cedar trees in the area. It's the easiest thing. A lot of the chemicals that industrial farms will use to prevent this, these fungicides, are also killing off the good fungus that's in the leaves. And we'll see that later when we start looking at like the functional ecology. Okay, moving on to our next group, unless you got more questions about this life cycle. It will be on the exam, but you will have access to all these slides and stuff for it.
I don't know if you can see the conversation.
Oh, no.
Christina has asked 2 questions.
I'm sorry.
She said, do all teleospores need winter before developing?
Yes.
And do rust change hosts, or do they always use the same ones for a certain species?
On like human lifespan, they always use the exact same hosts. Over the past 10,000 years, they've been using more or less the same hosts. But eventually, they do make a mistake. They land on the wrong host and are somehow able to get into it. And that's how we get new rust species, is one lands on, quote unquote, the wrong host and is able to survive well enough to produce some spermatia or some teleospores. And suddenly you've got a new species of rust that'll be diverging from the other ones. And so that's why we've got, you know, hundreds and thousands of different species of rusts. It's similar to, I don't know, if you took me for Bio 1620, we talked about cuckoo birds, right? And how we get new, like, gentus, new races of cuckoos is one accidentally lays an egg in the wrong nest and it doesn't get killed. So that's how we get new ones, new rust species also. All right. Now the oyster mushroom. Chandler put a photo of a really nice looking patch of oysters that she found up on our team's channel. They can take a lot of different colors. You can find pink ones and blue ones, white ones like this, but they all look like this, and they're all technically quite edible and delicious unless you're like accidentally deathly allergic to them. Okay. Oyster mushrooms are the ones that we're going to try to grow in this class. This will be a bonus assignment because it does involve some work. Okay. but it's actually not too hard. There's a lot of resources on the Hillbilly Mycology page. I posted links to a bunch of videos of other people on YouTube and even myself growing these, okay? You've got auger dishes already. Save a few of those. Go to the Asian market, buy some oyster mushrooms for $1.50, rip it open, and with clean pinchers, just pluck out a little piece and stick it on your dish. Presto, you've got yourself a culture of oyster mushrooms. It's really that easy. The videos will walk you through it. Then you just have to get that to grow on a bunch of straw and keep it in a bucket with a couple holes, and boom, you get oyster mushrooms. Okay. These are an agaricales. I spelled it wrong. It's not aragoricales. It's agaricales. Okay. So they're your typical guild mushrooms. The stem is off to the side, though. We tend to think of these as being saprotrophs, meaning they eat dead stuff. So out in the woods, you're going to find them largely on dead trees, fallen logs, that kind of thing. But they're also predatory. And so oyster mushrooms actually will like hunt out and kill nematodes in a rotten log also. Okay, so that's an agaricales. Here's another one, Lycoperdon. Chances are good that you'll see this one out in the woods also. It's just your standard puffball. Okay, it looks like it's covered in little dots. A baby one looks like this, fully grown looks like that. When it's fully developed, it'll be like lightweight, feel hollow, and when you touch it, it'll poof out a bunch of spores. Okay, this is one of those quote-unquote gastromyces fungi. Okay, the spores develop inside in what we call the gleba. It's just another fancy, stupid name for the hymenium. Hymenium's on the inside here. Okay, you can see it's making basidia, pretty normal-looking basidia. It just happens to be inside, and it depends on something else coming along. Okay, tremella here.
I got a question about that
Yeah.
last one.
Right.
So if you're out in the wilderness and you come across these and you step on one and you breathe it in, not realizing what you did, is it potentially dangerous to you, or is it?
I'm gonna say don't like put your face over it.
Right.
Inhaler. But yeah, you don't want to like puff this into your nose and breathe deeply. Getting into your lungs, these aren't gonna like live in your lungs and start growing or anything. They could irritate your sinuses or your lungs. 6% of people, and we'll see this more when we look at medical mycology real soon. 6% of people are like allergic. You'll get like itchy eyes, stuffy nose kind of stuff from fungal spores. But most of those allergies are actually in like the ascomycetes, like bread mold, that kind of thing, the black mold that grows in your basement or whatever. These, I think you're going to be fine. If you're puffing it and you get some in your nose, I've never even noticed it unless you're like real unlucky and have an allergy to it. There's not going to be a problem. That said, you don't want to have like 20 billion of these spores in your lungs because they will clog you up. Yeah.
So is it like a musty smell to it? Do you know, or is it?
Yeah, it's an earthy. I like the smell of it. It's kind of an earthy. The puffball, this lycoperdon, almost has like a weird soapy smell. It's kind of hard to describe. I would say it smells like a lycoperdon because nothing else quite smells like it. It's like a soapy, earthy smell. If you find them, you'll know what I'm talking about instantly, especially if you find a young one, because those are safer to smell than one that's like gonna put a billion spores in your lungs. Just kind of like bust open one of these little babies, and you can get it real close to your nose and smell it. It's got this slightly acrid, kind of soapy dirt smell.
Interesting. Okay.
Yeah. It'd make a decent cologne, probably. All right, and then tremella, we saw these. This is one of those jellies. Okay. This was like that yellow jelly. I've found these. I live right by Grove Creek Canyon in Pleasant Grove, and I found these all over the place there along the river. They get weird. I'm not going to require you to know this life cycle, but just be aware that not all Basidiomycetes have a standard life cycle. I guess you've already seen that with the rusts. It's super annoying. These are also pretty annoying because they've got a yeast stage. Okay, so they can go back in and out of this yeast stage also, moving to mycelial to yeast, but the fruiting body, the jelly that you see, that is the dikaryotic part. Nobody ever sees these in a yeast stage. We just noticed it in the lab when we were trying to grow these. Okay, so those are the main ones I wanted to chat about and introduce you to. Basidiomycetes are economically hugely important. Indigo agriculture right now, it's startup. I don't know if I didn't call it a startup anymore. It's 8 years old, but I think that company is now valued at like $8 billion, and they're always hiring mycologists to work on rusts, okay? And they're paying like 6 figures straight out of the bank because it's so economically important, okay? They're found all over the place. Basidiomycetes do a large amount of the carbon cycling. They're the only things that can break down lignin. So a tree falls over in the forest, it's not going to go back into the carbon cycle unless it's burned, which I guess is pretty likely here in Utah or anywhere in the western US. It can either be burned or it can be digested by Basidiomycetes. Okay, so they're super crucial for keeping CO2 in the air for our plants. Okay, they're important diseases, but also all those ectomycorrhizae that pine trees depend on. Okay, pine trees can't really grow without them. Those are Basidiomycetes. We've got a few animal pathogens, not really many. As far as human pathogens go, no. But as far as human poisons go, definitely yes. Okay. When we, let's see if I can pull this up. I want to introduce you real quick to some maybe common, very poisonous mushrooms that you're likely to find here. Amanita is the genus name. So you'll be familiar with the traditional crazy looking red mushroom with white spots. You'll find that here in Utah, but it's much more common. What is Phalloides? Oh yeah. You're gonna find this one underneath pine trees here in Utah. Looks like a little standard mushroom. It's got this like little veil on the stem. It's white, it's got nice white gills. You can pick it, you can touch it, but it's a wash your hands situation. So my recommendation is if you see a white mushroom with this ring around the stem, if you've got small kids at home or are planning on like licking your fingers anytime soon, probably pick a different mushroom for your collection, okay? This group of 3 mushrooms right here may only be 3 or 4 inches tall, maybe up to 6 inches tall. They get about this big. Easily be enough to kill everybody in our class. Okay. There is no cure for it other than liver transplant. So let's avoid, if it's got this ring around the stem and it's white, just go ahead and skip it. Here's what they look like when they're young. They kind of burst out of a little egg, okay? And underneath the gills will actually be covered by this little veil. You won't be able to see the gills when they're this young. That veil left over is what makes that little ring, okay? So look out for Amanita. Definitely don't eat. You will die. There are very few species of mushrooms that will kill you, but the ones that will definitely kill you. Okay, skip this, move on to a different species unless you're feeling brave. Okay, if you do have kids or pets at home, don't bring this into your house and stick it in your fridge. It's just not worth it. Pick a different one for your assignment.
For our assignment, can we just put these in like separate plastic bags or something for storing them until we get home?
I may show you. I've got on the website, if you go up to the top, the Hillbilly Mycology. Okay, and then go down to protocols, collecting and identifying macrofungi. All right, so these links here take you to some really pretty handy websites. So this will tell you where to look, what kind of notes to take, how to collect them. It's going to tell you to store them in a paper bag, not plastic. They'll rot if you keep them in a plastic bag. So you're going to stick them in a paper bag, bring them home, keep them in your fridge in a paper bag. Okay. Recommendations on how to collect macrofungi. So this is like saying a basket's actually more useful than a backpack because you don't crush them. Okay. It's going to say, hey, here's how to make a spore print. Okay, spore prints are really handy for identifying mushrooms because you can look at the color of the spores and even the shape of them with your little microscope. Okay, so look through these resources, and there's actually pretty good information on here for how to identify mushrooms, what characteristics to look for, what notes to take when you find it, right? Take a picture of it in place before you pluck it out. Cut one in half to look at where the gills are, that kind of thing. We've got the illustrated glossary of mushroom terms. So if you want to see like a real photo of some of these crazy terms, that'll take you there. Okay. And then the morphology illustrated guide, which you have to click on download right there. It'll take you to it. So this little section will get you through this assignment. I would like it if you made spore prints of all of these. That basically consists of taking a small piece of aluminum foil, cutting the cap off your mushroom, like cut the stem real close, and then just set the cap of your mushroom on top of the aluminum foil, stick a cup or bowl over it to keep the wind from blowing, wait overnight, and you're gonna get this cool pattern. I've got one here. But here on the website, it's nice instructions, some videos and stuff about how to make spore prints. Let me stop sharing my screen so I can show you this a bit bigger. So I've got a piece of aluminum foil here where I've been keeping a spore print. I don't know if you'll be able to see that. If that's working or not for you.
Yeah, that works.
Okay, yeah, I just set the mushroom cap down on this foil, stuck like a mug over it overnight, and I've got thousands of spores on there. Some people make artwork out of it. It's kind of fun. Those spores are going to help you identify a fungus. Definitely, you'll be able to see a color. Okay, if they're white, pink, cinnamon, brown colored, that all kind of goes into what you put on your paper to describe the fungus that you found. Okay. Any other questions? This is probably our most time-sensitive lab assignment because being Utah, we're just going to have a pretty short window. Once it starts snowing hard in the mountains, mushroom season's over. So do not save this one for December. Okay. If you're going to put off lab assignments, put off the other ones.
So how long do you think we have before we can't really do that one? Just time one. What do you think?
Whenever we get our first big snow in the mountains, that's pretty much the end.
Okay.
I don't know when that's going to be this year, but
Usually right before Halloween.
Yeah, I was going to say end of October, early November is probably when it's over.
So just try to get it done in the next couple weeks then.
Yeah, I would. Even into mid-October. Obviously, like lower elevations are going to start having mushrooms later than the high elevations. Right now, if you go, up on the benches or back into the valleys, then you're good right now. Especially with things like oyster mushrooms, those are really happy right now. So you'll find stuff now through the next month.
What are the mushrooms that you usually find in your grass in your front lawn or something? What are those ones?
Oh, chlorophyllum is the genus name of those. They're an agaric mushroom, so they're like in that monophyletic, like true euagaric clade. Common name for them is lawnmower mushroom, and they're responsible for the most poisonings of any other mushroom species in America every year. They apparently taste quite good, and so people will cook them up and eat them, and then spend the next roughly 36 hours with vomiting and diarrhea so hard that you will wind up vomiting blood.
So are those ones usually mistaken for the ones that you can just buy at the grocery store?
They look real similar, don't they? They look like a portobello. And so that's what happens. People see that and they think portobello, they eat it. Very rare that people will die from that. It's usually you'd have to have underlying health conditions, but I don't think it would be fun to vomit for 36 hours. Yeah, vomiting blood for 36 hours. So they won't kill you, but they'll make you wish you were dead.
Got it.
But it'd be a good collection, okay? Because they're easy to find, especially if you get like a light rain or something after mowing the lawn, they'll pop up.
Okay.
Yep.
All right. Thanks.
Mm-hmm. All right, we are out of time. I will try to stay in touch with you all over Teams. If you send me Canvas messages, some of you have, those take like a day or two to get to me before I get the notification from Canvas. So if you send me a message here on Teams, I get it instantly and can respond, so it's a lot faster. All right.
Dr. Zahn, can I ask you a couple of quick questions? Just
please.
About
Yeah.
The labs. So I might have missed that. I struggle with the online. I'm more of a visual learner. So as far as like submitting the lab stuff, so you say we have a slide. Do we need to go buy slides and then put them on, or do we have the slides to like put them on and take the picture?
Oh, I gave you one in your lab packet.
So we can just use the same slide over and over to...
Yeah, just give it a rinse in the sink, dry it off with a paper towel, and you're good to go. The only thing to be careful with is the cover slips. They're pretty fragile, and so just be careful when rinsing those. Yeah,
okay.
Just keep reusing it.
Okay, so we just take whatever, because I have all the things, I just haven't submitted any pictures of it, so just put it on there, take a picture of it, and then do we keep all of like the pollen? I know you said to keep the bread mold, not the bread itself, but just put it on a plate.
Yeah, once you get it growing on a plate, keep that. You can throw out your bread. The chytrids is another story. They're much harder to keep growing in a culture, and so we're not even going to bother trying with those.
And then with the Ascomycetes, so there's supposed to be that little line in between.
Mm-hmm.
So we just take a piece of whatever is on the line and just kind of smush it a little bit onto the slide.
Get a picture of it in place. So like just put your scope down on the surface of the auger. Don't crunch it in too far. and get a picture of it in place so you can kind of see it, how it grows normally, and then pluck one of those off and gently smash it on your slide so you can look at the ascospores. Yeah.
And do you want us to submit both of those, like the one that in place and then?
Yeah. Mm-hmm. And those instructions are on the Canvas assignment.
Okay. And you said, oh, go ahead.
Yeah.
Okay.
So it should just say kind of like what I'm looking for in a given assignment.
Okay. And then as far as identifying what's on there, because you mentioned that there'll probably be something like that on the test. So do we just need to like figure out on our own what we're looking at, or how can we sort of identify?
Yeah, and you should have the skills. I'm just looking for the terms, right? Parathecium, ascospore, ascus, mycelium, that kind of thing. You should know what part of the fungus you're seeing.
Okay, cool. Thank you.
I'm never going to have you identify species in an exam, okay? But being able to name the parts of what you're seeing is the important thing.
Thank you.
Mm-hmm.
I had a question about the oyster mushrooms.
Yeah.
So you said that we're gonna, for extra credit, right? If we can go down and buy one, and then we take it, cut it, and then takes part of it and put it in an auger dish, right?
Mm-hmm.
Wait till that grows.
Yep.
Right. And then do we transfer that to something else? I missed that part.
Yeah. I didn't walk you through it here today because I made videos for the whole thing.
Oh, okay.
So that whole process, it is, let me share my screen again. It's also on the Hillbilly Mycology page. So it's at the very bottom, grow oyster mushrooms at home. Okay, so Here's the overview of the steps. Here's how to find yourself a clone for free. That's the Asian market. Okay, it's like $2. And then how to transfer your culture to a new growth medium, how to make sure the culture is clean. Contamination is the problem here, and that's why this is bonus, because you really have to be careful about keeping things clean. So this will walk you through the whole process.
Okay.
Yep.
Thanks.
And then if we got time, I think we're gonna do some edible mycology. I can give tempeh cultures, these rhizopus. I can leave those at the lab for anyone interested in making tempeh, or make your own soy sauce, that kind of stuff. That's mycology right there. So we'll see what we can do with that. All right. I'll see you guys next time.
Wait.
Oh, wait.
Okay.
Thanks.
What? One random question. So, on my backup bread mold that I have, I'm getting these rather large for sporangia, and they're yellow. Do you have any idea what I?
No way.
Yeah.
Let's see here. Like this kind of?
Okay. So it's like that, except the black balls are yellow. Kind of. But no, it's not. No. They're not little ones that, because those are the ones that shoot out, right?
Right. Yeah. They're typically found in dung. They'll grow in bread. If you had cow poop in your soil sample, there's a good chance that you would get these.
Let me go grab it real quick.
Okay.
And here, let me turn my camera on, get some good lighting going, and maybe I can put it up to the camera here.
Okay.
Okay.
Right there. Hold it still. Let it.
You can see they're like yellow dots in the top left there. If I can.
Yeah. Man, it's tough with this, like.
The crappy camera.
Yeah.
but essentially it just looks like the normal stuff I had before, where it was white mycelium and then black sporangia, except they're yellow, and they're like twice as big as the black ones I was having.
Okay. See if you can get a couple microscope pictures of them. That's where I'm going to be able to tell what's going on.
All right, we'll do. I'll probably just send it to like the this page's chat.
Yeah, perfect.
All right, that's it. Thanks.
All right, later.